Okay, so welcome everyone to the third ACIP webinar on modeling and simulation. A little bit of background. ACIP has a strong history of using modeling and simulation as a tool in both basic science, but also in applied applications such as optimization or automation of processes. And yeah, we have been using this webinar as a way to showcase the capabilities at ACIP and also its partnering universities and companies. Yeah, and today it's my pleasure to have speakers from Technical University Graz and from Boku University that will talk about sustainability in bioprocessing. Yeah, so we have Barbara Petschacher and Peter Satzer. A little bit of intro to how the webinar is structured. So each presenter has 20 minutes for the presentation, followed by about 10 minutes of Q&A. As you can see, the webinar is recorded, and therefore, if you want, your questions can also be asked anonymously during the Q&A function you see at the bottom. We will stop recording after the first hour, and you can continue the discussion if there is interest. We will also offer the opportunity to switch to another Zoom channel where we can have breakout rooms if you want to have a face-to-face discussion with one of our speakers today. Yeah. So with that, let me introduce our first speaker. Yeah, Dr. Petschacher is a biotechnologist by training with a passion for sustainability topics, and she has been ACIP's Chief Sustainability Officer since last year after a strong involvement on sustainability research and teaching at Technical University Graz. With that, the stage is yours, Barbara. Thank you for coming today.
Thank you very much. Check if did I manage to share my screen?
Yeah, looks
okay,
good.
perfect.
Yeah, thank you.
Okay, so hello everyone. Thanks for joining the webinar. I am happy to talk today about sustainability assessment in bioprocesses, a little bit more on maybe on the general level, and then give some insights into hotspot analysis for glucosyl glycerol production, which was part of an EU project that I was involved in. So I suppose I'm not sure about the background of the audience today. So I really start with the basics, with sustainability development, sustainable development definition, as it was done in the Brundtland report already in 1987. And there it says that sustainable development is a development that meets the needs of the present without compromising the ability of future generations to meet their own needs. And probably most of you know that this is not only based on environmental sustainability, but that there are three pillars involved, and this also involves the economic sustainability and societal sustainability. And if we look at possibilities for sustainability assessment, methods that probably many of you already have heard about for the environmental assessment can be green metrics or life cycle assessment. For the economy, techno-economical assessment or life cycle costing. And for social sustainability, there is also a social life cycle assessment. And if all three of them are applied in a comprehensive approach, then we have the term of life cycle sustainability assessment, which then really takes into account all three pillars, just to clarify the nomenclature here. But today, I will really focus on the environmental sustainability. So why are we doing sustainability assessment? Of course, one reason that probably most of you think about already in the first place is because we really heavily already transgress now the planetary boundaries. We have crossed 6 of them. And if we want to counteract these problems, it really is necessary that we know what we are doing and that we know where we have the most impact to really mitigate these problems. But of course, sustainability assessment also has a lot of opportunities in it, because with sustainability assessment, we can identify improvement possibilities for products and processes. And this might lead us to avoid later on regrets, because we can build for future-proof innovations. And another reason to do this might also be to meet expectations from industry and regulation. Industry, especially large industry, is already heavily involved in sustainability assessment in their daily life, because also the CSRD, so the Corporate Sustainability Reporting Directive, has become active, went into force this year, and will also involve smaller companies in the next years. So these activities on trying to evaluate as well as the organization, as also then the products, as we can see for like activities for the digital product passport, we will stay with us for the next years for sure. Another important topic is to solve problems without creating new ones. And why do we want to do early on already assessment, sustainability assessment, is because if we already take decisions or early on in the design process, and these are locked then very early on, and then it doesn't make sense too much to evaluate the sustainability in a ex post evaluation. We really need to do this early on. So how is sustainability assessment done? I want to only shortly give you an overview here from the kind of more qualitative and maybe a little bit easier to do principles in the upper part of the table, to the more comprehensive and also more difficult to do in the end. We can start with green chemistry or eco-scale, which is more qualitative to do, to check if you adhere to certain principles that should be followed. Then in the middle we have this part of the green metrics. There are of course more. This is not a full list, and I think Peter will more focus on this part later on. I maybe only want to point out that they are more easily to achieve, because they often concentrate on masses. For the E factor, which normally kind of gives a value for how much waste is produced per product, there can also be an extension for the I just changed to the pointer, for the energy carbon dioxide to include this. Sorry. So I just shortly want to give an outline of how life cycle assessment frameworks normally look like, because I'm not sure if everyone of you is aware of this. You start normally with a goal and scope definition, which I personally think is a very important part, because you really have to be aware yourself why you are doing it, with which also resources are you doing it, with which methodologies you have available, and especially also the system boundaries are of importance. In principle, of course, as the name says, life cycle analysis should be done over the whole life cycle, but sometimes you just don't have the possibility to have the data for things that happen outside of your gate, and maybe it is not necessary because you want to do a hotspot analysis, for example. Then we have the part of the inventory analysis, which includes the data collection, relating data to functional unit, to unit processes, the impact assessment part, which really then calculates from your inventory, from your data, the impacts that you have with your product or processes or organizations. And this is included in frameworks or in methodologies that really then kind of are integrated in maybe software solutions already. So this is kind of the action area of real LCA specialists to optimize then really the methods themselves. Normally you can just apply them. And then in the end, we have the interpretation phase, which also deals with uncertainties and sensitivities, and in the end wants to give recommendations maybe for the improvement of the process. There are guidelines around for these LCA frameworks. So, for example, the product environmental footprint from the which is promoted by the EU. Here you have also a list of the impact categories that are addressed in this methodology. If you only choose to do it with one impact category, this could, for example, be the carbon footprint. There also are ISO norms and methodologies available, as well as also from some sectoral clusters, like here for chemicals. And of course, there are quite some challenges associated with sustainability assessment in general. I kept here the structure of the life cycle assessment, but this is also true for all kinds of sustainability assessment. An important question, as I said before, is to choose the appropriate system boundaries. Here also one critical topic is multifunctionality, which means that you could have not only one product, but several products, and then have to decide how you allocate your burdens to the different products. In the impact assessment, sorry, there's somehow something went wrong here now. I duplicated one. Unfortunately, I obviously missed out the most important part here by copying in the something in the inventory. There we really have the most challenges, because it is really important to get to good data, and this is really often not so easy, because the data availability is really often not quite scarce. And for example, especially in chemicals background, there are really a lot of data gaps where still things have to be evaluated, and we will hear also later from Peter how we can deal with this. Another problem here is also the question of the scale. As we said, we maybe want to do prospective assessment, and do this at the stage where we only have data from lab scale, so from low technology readiness levels, and want to go further on into higher scales without having the data available. I will have a slide on that in the next second. And in the interpretation phase, we have to be aware that there are quite strict also rules for example, if we want to compare results, because often, of course, one would like to state something like my product is better, or something like another product, but this really is only possible according to the standards if you really use the same methodology for evaluating, which in principle would mean that you have to do it yourself for both parts to compare, and this is often really not easy. And in this case, you would also need an external review for your assessment. So just to be aware, it's very important that you align what you expect from your assessment to the goal that you have in your assessment. And to just shortly dig a little bit deeper into this prospective assessment problematics. Don't be afraid of this graph, which I took from Niels Tonemann paper. I just want to point out here that we are maybe sometimes not aware of all the problems that come along with prospective assessment, and we cannot solve them probably all at the moment. And I really hope that with the time and with people going more into details of scenario analysis and so on, this will get easier. But the problem that we as engineers often mostly are concentrated on are if we have an emerging technology at laboratory level, then we think about scaling up to a not existing yet higher scale. So this is one of the challenges that we have to meet. But on the other side, there's also the time challenge, because if we develop now a methodology that should come into market, let's say in 5 years, then we also probably can await that technologies also in the background systems or systems already develop in this time. So for example, if we apply an electricity mix that we have today, this probably will not be the same in 5 years. And then it might be that we compare things that don't fit together. But for me, I would say that this scenario analysis and so on then really is the domain of real LCA methodology experts that hopefully will help us to have here scenarios ready that we can use without having to really assess this on our own. But on the what for sure is often encountered is the upscaling in the process line. And here we have different possibilities how that can be done. We have the process simulation, which is kind of the most comprehensive variant if we can use as programs as ASP and SuperPro Designer. But for this, we have to have quite some knowledge about our process that we later on want to run. We can do manual calculations or based on physical equations. We can use molecular structure models, which Peter also will mention later on, which means that we have at least the structure maybe of our substances available. And in many cases, we just use proxies, which means we know similar processes, we know expert knowledge from somewhere, we know maybe instrument manuals, and can derive the data that we need from this. And it's mainly the energy data that is really difficult to assess. The mass balances are easier because they are linear, can be linearly scaled normally, but the energy really, of course, you need less energy if you get to larger scales compared to the laboratory scales. And if you want to dig into this a little bit more, there's a nice paper from Zoe et al., which gives you kind of a decision tree a little bit to see which kind of upscaling method might be applicable in your case, and with some references to check how other people did it. Okay, now I told you a lot about how complex sustainability assessment is, but now I want to kind of step back to just doing it, which I really think is an important first step, and not to get annoyed because of all these uncertainties that still are around. And so I want to show you an example how we did it in this Carbafin EU project. Here we dealt with a biocatalytic process, which was about producing glucosyl glycerol, which can be used as a functional ingredient for cosmetic industries. And here the used biocatalyst is a sucrose phosphorylase, which transfers the glucose moiety from a sucrose to glycerol, and fructose is a byproduct. So the group of Bernd Nedetzky and Christiane Lula has done a lot of research already before on this reaction. So we were able to already achieve here an innovation action project in the EU Horizon 2020 program. And this made it possible to really use demonstration level data partly already, which was nice. We did a hotspot analysis, so this was the aim of the study, to get to have an internal to develop an internal improvement plan. We only used cradle to gate. The functional unit was 1 kilogram with a reference stream of 1,000 kilogram, which was a batch for the company. And And in this case, it was a continuous conversion with the enzyme coming from an external supplier. And in the start of the project, the downstream processing was chromatography based. And as I said before, the foreground data partly came from lab scale, partly from demo scale. We calculated it manually. We used proxies for gaps. We did not include the transport. So for sure, this was not the perfect LCA to be done with a lot of gaps, but still I think it shows how important it is to just do it and get a first impression of where you have your hotspots. I will not go into very detail here. We used the ecological footprint, which also is a single impact category parameter, which kind of gives you the area that you need to embed a whole life cycle sustainably in the biosphere. And this is was developed at TU Graz several decades ago already, and is used by our company partner Strateco. And with the same method, but you can also do a greenhouse gas emission footprint. So what we saw is that maybe not unexpected, but in the original process, the chromatography step really was the hotspot. And this accounted for about 30% of the overall environmental footprint. And we then, our company partner within the project changed to a nanofiltration-based downstream processing, and by this could reduce the footprint by 25%. We later on then also looked on further hotspots for the improvement plan. And here we see that, for example, when we look at the first step, which we call substrate preparation, which mainly includes the raw materials, we saw that glycerol was used in a high molar excess over sucrose. And the improvement suggestion here was to do better, so to increase reaction and enzyme engineering to be able to run the reaction with lower molar excess of glycerol. And this was really followed up also in the project. And I can recommend to look at the papers of the Nidetsky group here in more detail. Another important point, as we can see here with the blue sectors number 3, which refer to the electricity, is of course that a change to renewable electricity remarkably reduces the footprint. Then the enzyme solution, which is the number 4 here, as I said before, was here in this case, the data was taken from a database, so it was not own data. And in this case, I would recommend to really check also again if maybe better own data is available, just to make sure if the impact really is as high as it is shown here. And another possibility would also be to change the acid, as the hydrochloric acid has a larger impact than the nitric acid. Also for the reverse of osmosis water, the influence of the energy is very high. And one interesting part, which we maybe did not think about in the beginning, was that the impact of the vessels where the product was filled was quite high in the overall impact, because it was filled in 1-liter vessels. And so the part of the plastic is not too small in the overall impact. So in the end, if we would apply all these or several of these improvements, the footprint could be further reduced to 49% of the one before. So yeah, and finally, I just want to point out that as outcome of this project, we also went into a partnership with Strateco, because what we saw is that it is very important to have, or we found it very important to have the really a good tool or method available how to do the data collection and the assessment in one, and maybe with avoiding to have 3 different Excels where you can always do a mistake when you deal with a lot of data. So what we designed here is an inventory collection and assessment tool in one, where you can really have also your goal and scope description. You have the flow chart in, you can put even process information like titers and so on, some data quality, hotspot analysis, and can do the hotspot analysis, scenario analysis, and have a fast visualization because this is already prepared in the tool. Exactly. Sorry, I forgot to click. And we have also a report template available. And we would be happy if anyone would want to collaborate with us on some sustainability investments, in investigations, then we would be happy to do that. I'm a little bit behind time. Should I skip this or should I still go on for the last 2 slides?
I mean, okay, try and come to an end soon, but
Yeah.
please don't skip the slide.
I just put some remarks from my own here. So I really think that the sustainability assessment will stay, and that it will be increasingly important. That it also we from the research side really should get used to include this in our daily work. But of course, to make it not so cumbersome and more comparable, it needs to be better defined how to do it, also in the lower technology readiness levels. But initiatives as Safe and Sustainable by Design guidelines are trying to do exactly this at the moment, so I hope that this will go on. I also think that digitalization will have a major impact in the future, which we will see how that will develop. I see a little bit the danger that we have too little high quality background data if we don't still stick on really doing that in a thorough manner. I think that the database access, which of course curation of data needs some work, and this will always cost some money. And this can either be done by paying it from project money, or it might be publicly funded to have open access. We will see this. And I really think that the sustainability assessment will help to deliver better and often cheaper products and processes. So my take-home message is start doing it. Almost no matter how, but just start doing it, because it might already be worth to just think about sustainability in your processes, but later strive to improve, because if you stay at the beginner level, then probably it might be not enough. And here's only just some information on ACIP activities like our tech offers, or also a nice podcast that you could also look at. So thank you very much for your attention, and I'm sorry for taking away the time for questions.
Thank you, Paula. A very interesting talk. We actually already have a question from the chat, but everyone feel free to ask questions via the Q&A feature, or raise your hand if you want to ask a question live. We have a question from Adriana Diaz-Triana. The question is, good afternoon. May I ask if there were any product category rules used for this assessment? And how do you consider variation in the reaction yield?
Variation in the?
Reaction yield.
Reaction yield. The product category rule was, so it was really this very kind of special sustainable process index, which is not in an assessment as it is in any of the product categories from the EU, and we were also not in one of these product category rule schemes here. So we just did it from scratch, let's say, in the beginning. At that time, I'm not even sure if, so it's some years past now, I'm not even sure if there was already any product category rule already developed really at that time. sorry. So, and for the deviation in the product yield, it was the process was developed already very good, and it was, so I think our partner did 2 parallel demonstration runs, and we chose finally one, but I think not really because the yields varied too much, but because of there were some other problems. But of course, I know that if you have different yields from several runs, then of course this has a big impact on the sustainability outcome, because the kilograms vary then in each case. I'm not sure if I answered the question by this, but
yeah, I hope this answers the questions. Are there any more questions? So I was wondering, like, so this is biotech specific, the inventory that's kind of lacking, or is this a general problem? Like, are there industries that have a better inventory?
I think it's not biotech specific, but maybe chemical specific, because we have if you have a lot of so I think you run into this problem whenever you have a lot of impact, a lot of input parameters, like for example in a fermentation where you have a lot of different materials that go into it. I think that, for example, in agriculture, there might be a little bit better data availability, but even there, it might depend also where you put the cutoff, because I suppose if you look for every single material that you need there, it probably also is not much easier. So this is, I would say, it's more a general problem, but specifically all the chemicals need still some evaluation.
Something we could learn from other industries, I guess, or to improve our situation. But okay, with that, yeah, since there are no more questions, thank you, Barbara, again.
Thank you.
There will be opportunity to ask more questions. So if you have any, type them in, and Barbara can type an answer. So the Q&A tool is open the whole time. And then, yeah, there will be opportunity for one-on-one discussions afterwards anyways. We will post a link to a breakout room then in the chat. So let me introduce our next speaker. Dr. Satze is a biotechnologist specializing in bioprocess engineering and sustainable manufacturing. And alongside his academic roles here at BOCO and ACIP, he also co-founded P4B GmbH, a startup developing AI-driven 3D printed bioreactors to develop biopharmaceutical development and minimize resource use. With that, I hand over to you, Peter. Thank you.
Thank you very much. I have to move that out of the way. Okay. So I will be talking more about bioprocess sustainability, so bioprocess modeling. And I think we need to look at the bioprocess as a whole, and Barbara already mentioned it, all the materials that are going into the process and that we might not have enough data for it actually. But for the whole bioprocess, we have chemicals and solvents going into the bioprocess, plastics and instrumentation. Barbara also mentioned that already. We have the waste streams going out, but also we have the whole support structure and overheads. And the thing is that we have to look at also not only inside and outside the box, so what's going in and out of the box, but also the box itself, because there have been a couple of white papers actually on the clean room energy consumption. And the clean room energy consumption might actually be the largest contributor to our facility or to our energy needs in the facility, being up to 2/3 of the actual energy used in the whole facility. So we need to keep in mind that we are not only running a process, but we're running a process in a certain space. But what do we actually need for process modeling? It's quite simple in principle, but hard to get the data. It's what goes into the process, how much goes into the process, and how long. And for that, we need a process definition. It's basically, we need to know what we are going to do. But in very early stages in the process development, that might not be that easy. How that could look like, and I will come back to that example because I have some data on that later in the talk, is, for instance, a proteinic chromatography. You might know how much column volumes you need for each of the steps. You might know what kind of buffer you intend to use. But you might not know yet at the moment of, okay, what's the sustainability impact of if I'm losing less or more of the buffer or a different buffer species. And that's one of the topics that we are targeting with Boehringer Ingelheim on assessing what the actual buffer species and buffer impact is on chromatography. So how do you actually start? PMI was already mentioned. We need key performance indicators. We need some kind of measurement to know how good we are in the process. And there's the process mass intensity. There's also something called Varian that actually me and a student introduced. It's called the water-related impact of energy. It's the energy associated with the water that you actually use in the process. And for sure, we need additional KPIs in the future that we need to develop and formulate to characterize our processes. So where does the PMI actually come from? It actually comes from synthesis. And for synthesis, it makes sense. So how much material do I actually put into the process and how much do I actually get out of my as a product? For biopharmaceuticals, it has some problems because our processes are largely water-driven. We have everything is highly diluted in comparison to any chemical process. So the PMI is also very water-dominated if you use that as a key performance indicator. So we introduced a Varian to at least have the energy associated with that water that goes into the process in our modeling as well. So that's an example from a biopharmaceutical process. Total input materials, total output materials, classical definition of the process mass intensity. And you see 95% of what we are actually going to put into the process is water. That's why PMI might not be the right thing to do for a biopharmaceutical process or a bioprocess where you don't have highly concentrated feedstock solutions. So PMI doesn't really measure the absolute environmental footprint. It rather measures the material efficiency of your process, which is an issue with most of your materials that are going in as water. You also see on the lower side here distributions around different unit ops. And you see chromatography sticks out a lot, which means there we have the highest water consumption in any of the process steps that we have in a pharmaceutical process. So that's also why many companies are now concentrating on, let's fix that first before going to anything else. The PMI, of course, is titer dependent. And you see here on the lower side a graph of cell culture titer versus total PMI of a process, but it also varies a lot and it's not necessarily size dependent. So it's not necessarily that your PMI might go down during scale up. That's usually not happening, which means you have to fix it already at lab scale and cannot hope for any scale up effects on that front, which also makes sense. I mean, the PMI is just the mass material that you use. It can be somewhat scale dependent, or some unit ops can be scale dependent just because they are actually oversized for small processes. You see here an example for one of the publications of ours. It's the PMI of a centrifugation step of monoclonal antibody production for cell removal. And the huge bar here is actually the cleaning of the centrifuge. So if you have a lot of usage of water in some unit step that's actually not associated with the process itself, but for preparation and cleaning, there might be scale effects on that that are actually substantial. Now to the thing of, okay, we're now having all this water use, but how do we actually relate that to an energy or CO2 impact? And that's where we went with the water-related impact of energy as a key performance indicator. That now all sounds like it's complicated and it looks complicated and the formula looks complicated, but it's actually just the process mass intensity of your water multiplied by the actual CO2 equivalent that you need to produce that water. And that CO2 equivalent, of course, is different for process water than for water for injection, because water for injection needs more energy to be more clean to be actually used in a biopharmaceutical process. So what's now the main driver of this energy associated with the water that you're using? And the biggest contributor is the clean steam in your bioprocess, which means it's actually the sterilization of your whole equipment that you have in the bioprocess that has your largest impact. That's also why you need to have a kind of a holistic view of the bioprocess. If you would only do the materials that you're going in for the actual purification or for the actual use of the equipment in the process, and do not include cleaning before and after, you might miss the main contributors to your CO2 emission for your process. So you need to include that, even if you're doing then only a cradle to gate and see, okay, I have my product finished, I'm not caring about waste management, but you need to include that because otherwise you're missing maybe the main portion of the contributors to your process. We also did an analysis in that project on hot versus cold WFI, because that's a hot discussion point, or has been at least 5 years ago. The total impact is not as big as we hoped for. So by switching from a hot to cold WFI production, we only saw a 10 to 15% impact on the complete process in reduction of CO2. It's not nothing, of course. I mean, take it if you can. But it's not the 50 or more % that we hoped for. The Varian is scale dependent, but also single-use and stainless steel dependent. So the mode of operation that you're using, of course, I've talked about the main contributor being the clean steam, sterilization steam that you use. And that's, of course, used in stainless steel operation. For single use, you're throwing the plastic away. And there's always be the discussion, is it better to throw plastic away, or is it better to clean your stainless steel reactor? I would say it's still a little bit open for debate. I mean, we saw benefits for the single use. Of course, the single-use manufacturers see benefits for the single-use equipment as well. I mean, sure they do. Yes, just be aware that in one case you have to deal with the plastics and have to deal with the CO2 emission for the production and incineration of the plastics. And in the other case, you have to deal with the clean steam production and the energy needed to heat up your stainless steel reactor for sterilization. Both need to be accounted for, because otherwise you're just calculating nothing of relevance if you're not having the whole picture there. One important point you see on the right-hand bottom side, the water-related cost per kilogram of active pharmaceutical ingredient. And it correlates with the Varian, which means sustainability is nothing that costs you something, it saves money. If you're using less energy, you're saving money. If you're using less materials, you're saving money. So it's not only we're doing that for the benefit of the climate. Of course, we're doing that. That's why we started maybe. But if you need some argument for your management of why they should care, it can bring down cost if you're using less materials, if you're using less energy. And of course, at some point, we will all pay a higher cost for CO2 emissions as well. Which means the cost pressure will only increase in the future. I've talked a little bit about lab space and how that could be a large contributor. So on the right-hand side, you see another kind of white paper where they reported 60% energy use is due to the HVAC system. So that means ventilation and climatization of the lab space. That's a huge contributor, and it's often overlooked. But software packages such as the already mentioned SuperPro Designer and the Biosolve can at least give you the floor space use. And I know that Biosolve is also working in implementing the sustainability impact of lab space into their software. We're not affiliated with them, but I've worked with the software and it gives you good results. Here you see some analysis of different floor space requirements and different clean room classes. You have to be aware that different clean room classes, of course, need different kind of energy to function, because you have higher volumetric exchange rates of the air for higher clean room classes. Which also means if you're thinking about going maybe continuous, maybe going fully closed system, maybe needing to use lower class clean room for your fully closed system, that also has sustainability impacts that you can claim if you want to go that route. What you see here, for instance, I've just marked that because there you have a stark difference. One of them is single use, one of them is stainless steel. And you not only see the benefits maybe that I've showed before of switching to single use for the actual energy that's used for the process materials or for the cleaning and sterilization, but you might also need less space. And if you need less space, that also means less energy and more sustainability of your process. So keep in mind that shrinking your process might be a substantial way to contribute to sustainability, just because you need less space and you need less clean room to maintain. That's also another example. Of course, floor space associated CO2 emissions are scale dependent, which means on larger scale, you need less floor space for the same amount of API that you're producing. Just to have some kind of context of the magnitude, you see here on the lower left side, lab space emissions could be as high as 400 kilograms CO2 equivalent per kilogram of API. And the variant that I had before was around 90. So which means, yes, we introduced the variant as a key performance indicator, but we need to take into account the lab space as well, because otherwise we might be again missing a large portion of the actual environmental impact of your process. So now an example of sustainability impact of materials. I mean, we've dealt now with the water and the lab space, but what about the buffers that I introduced in the beginning for the protein A chromatography, for instance? We started that project with Boehringer because, quite frankly, we had no idea what the impact was. Was it significant or not? And we ran into the first problem. Barbara talked about life cycle inventories, and we were using EcoInvent. And you see here on the left side some buffers that you typically encounter in chromatography, and everything that's red is not in EcoInvent. And EcoInvent is the most broad life cycle inventory that we have at the moment, which means a lot of the things that we deal with, we just don't have any data for. There is at least some way around it. It has been published by the ETH Zurich, which is called FineChem. It's a model that tries to correlate the chemical structure of chemical compounds with their CO2 impact. On the right lower side, you see the correlation between the predicted carbon footprint of a structure and the actual carbon footprint of a structure. And I would call that quite okay for what we are usually dealing with in terms of models. It is available as an R package, so you can implement that into your like framework if you're missing some data on chemical compounds. That's what we did for the things that we were missing here for buffer substances. Be aware that it's only an estimation, and you see here on the right-hand side the confidence of the model of FineChem on the actual value with a standard deviation, and you see it sometimes just includes 0 as well. Those models aren't very good, so don't take the values just like they are, but be aware that they come with a high uncertainty. But unfortunately, it's the best that we have. So now the question was, okay, we have the water, but how much CO2 is actually emitted in addition for the buffer substances? You see here on the right upper side, freshwater consumption, on the lower side, carbon footprint of the water itself in blue, and the buffering substances on top. And you see for the water, 50% of the net freshwater use is due to citrate in a 50 millimolar citrate buffer, which means a larger portion of water consumption in your process is actually not happening in your facility, but is happening for the production of the buffer substance that you're using in your facility. And for CO2 footprint, it's even more drastic because there 80–90% of your CO2 emission of the buffer is actually the buffer substance. So you have to have those materials in your process simulation and process modeling for ecological impact, because otherwise, again, you're missing a large portion of what you're actually dealing with. But that also means you get an additional point where you can actually optimize. And we did a calculation for a full chromatographic step, like the one that I introduced at the beginning. Protein A chromatography of a monoclonal antibody, quite standard. Equilibration, wash solution, cleaning in place, and your total water consumption and your total carbon footprint of the process. And you see, by just changing your elution from one buffer to a different buffer, you could maybe save 50% of your total carbon footprint of the buffers that go into your system. Of course, the buffers that go into the system are not your whole CO2 footprint, but it's a major part. Of course, you then, I mean, we have introduced the variant, so we ought to use it, right? So we introduced the variant in that calculation as well to relate the water that we are using. And you see here still 50%, around 50%, regardless of which WFI production you're using, of CO2 impact is due to your buffering substance. And the rest is due to the water, the WFI that you're actually introducing. You also see that everything that's more mineral-based buffer substance like phosphate, on the left-hand side you have the phosphate buffered saline, has a significantly lower CO2 footprint than any organic buffer. Let's call it organic buffer. It's in this case a citrate buffer. And then in the end, if you're introducing the variant into our total calculation as well, you still see, just by switching elution buffer in the protein A, 20% of savings of your CO2 footprint. And on the right-hand side, I also brought some data for cation exchange chromatography, another standard step in antibody purification. And you see 30% can be saved in CO2 footprint if you're changing from one buffer to the next. And at least when I talk to bioprocess specialists, they weren't aware that switching from citrate to acetate buffer could actually have that huge of an impact on your total sustainability. And that, in my opinion, highlights how important it is to take all material flows into account, and why I think that the process mass intensity just isn't enough for bioprocesses. And with that, I want to thank the project partner that we had. So the first data that I showed was from Project Next Biofarm, funded by the Horizon 2020 project, and with a quite large network of industrial partners. And the data on chromatography that I showed now lasts is about sustainable chromatography project that we are having with Boehringer, which is about understanding the ecological impact of chromatographic buffers. And let's work on a sustainable future together, maybe through ACIP, maybe through POCO. Thank you.
Thank you, Peter. Really interesting presentation. Yeah, we actually already have a question from the audience there, but everyone else, feel free to ask more questions there or raise your hand. Martin Bergholt asks, could you actually elaborate a little bit further about the model used in order to calculate the FinChem data for chemicals?
Yeah, so the FinChem model, it's not from our group, it's from the ETH Zurich. What you're doing is they're using a machine learning approach where they try to dissect the structure into different components. Like, okay, I'm having a ring, I'm having some additional structure. I've read the paper. It's a multi-headed machine learning approach. It's basically trying to correlate the structure with the CO2 emission data that's found in EcoInvent. So it's again based on EcoInvent and EcoInvent data. And the model tries to relate the structure to the CO2 impact. There is no process data in there. There is no like fundamental, like, okay, you have this kind of structure, you need that kind of energy or whatever. It's purely correlation, like structure correlation to CO2 data correlation.
Okay, I hope this answers that question. We have another one, but first I wanted to to point out, I'm still surprised that in this EcoInvent list, we don't actually have data for trees, which coming from biotech. It's such a basic buffer compound, right? But it really shows those inventories are not focused on our field. So
no, those are coming from agricultural, and though there have been huge changes there as well. So it's like, okay, they have been learning all the time as well, and models are refined, because some of the data in EcoInvent is actually not primary quality data, it's also modeled data. So we are kind of partially modeling on modeled data, which is dangerous at the end, because we need to find some way of getting better quality data. But it's scarce, especially for biopharmaceutical. Also, the citrate that we have been using in that modeling is not biopharmaceutical grade citrate.
Okay.
There is no data on that. It's purely if you would bought it off the shelf as food ingredient.
Okay. So on to the next question. We have an anonymous one. Have you ever evaluated alternatives to chromatography methods, like magnetic separation, even in process scale?
No, we have not. I mean, if you want to, we are of course open to it, and we can discuss the project or the process definition and see that we model that. It can be done if we have all the process definitions, but we haven't done it yet.
Okay, and then we have a follow-up question to the FineChem one. Any toxicology data that is considered?
No. FineChem is CO2 only. It's not even water impact. So we started out with, okay, we want to have like a model and a estimation of the water consumption to produce certain chemical substances as well. It's not in there. It's CO2 only, unfortunately.
Okay. And then one last question from me to both of you, actually. So what is the best way to get started? Which are the guidelines to follow if I want to? I mean, I guess reaching out to you would be my first step. Is there like one place to start, or does it depend a lot on what you are doing?
I think sustainability encompasses a couple of different activities, like sustainability modeling, coming from a process modeling perspective like I do. I think, okay, reach out to me, perfectly fine. I'm happy to help. But actually for us, usually the first step is process definition. You need to know what you're doing, what materials go in and what materials go out. Without that, you cannot start any calculation on what impact that might have. And it's sometimes surprisingly difficult to get that compiled.
I also think that, I mean, it's a principled decision, maybe if you stay kind with the more green metric based things where you can go quiet for some time with the masses, which is good. Of course, I completely agree with Peter that you need to do the inventory and the process description anyway first. When I started with the LCA things, I like to look at the LCA guidelines, although I did not really apply them, but it helps so much to see what questions one really should think about and to define. I also think it is probably important to know how much time and money you have available and to which kind of assessment that really fits in the end, because I mean you can do LCA studies probably for years to get really nice results. And I think for me this is really still a very crucial and not easy to answer question. I hope to a certain extent that these toolboxes, like they are developed, for example, for the safe and sustainable by design guidelines, which by the way also then should include toxicity related aspects. And there are these bigger EU projects like the PARC running, I think, which they try to develop toolboxes that one then easily could apply, but I'm not up to date how close we are to a list of what could be done.
Okay. Great. With that, we are out of time.
I think, one more question.
There's one
new
follow-up question. Yeah.
Fantastic. Renate Photo, thank you for the excellent presentation. Is there any difference in water consumption and or CO2 footprint, whether citrate is generated via chemical synthesis or via microorganism in a bioreactor?
I'm pretty sure there is, but I don't have any data on it. That's the whole issue that we are running into all the time. So EcoInvent doesn't distinguish between the two. At least I don't know that they're having two different data sets on that in EcoInvent. I think the one that is in EcoInvent is microorganisms in a bioreactor. So issue again is data availability. If there is some data on that, we can include that into analysis. The issue is with feeding that data, you would have to make a whole LCA for citrate production. And at least for us, that was not the goal of any project.
And what I also sometimes encounter it is that there might be some literature studies, also some literature on some LCA on some compounds, but if you look at this, there are now quite some studies that do on a meta-analysis level look at LCA studies in literature, and what you always see that in many cases, which is completely understandable, of course, but in most cases, they are not then really done in a standardized way, and that prohibits that they finally end up in EcoInvent, because this really tries to be kind of a high-level data, and if then the analysis that are there are not done in a standardized way, then you cannot include them, and additionally probably is also work again to prepare data sets in a way that they can be used in EcoInvent, and that also is something that probably not so often is financed then in the end. But yeah, maybe one could look if there's a literature study on citrate done like this and like that, but then it's still not in the database, even if it would be in the literature. But it could maybe give a hint, but I have no idea if there is one.
Okay. With that, we are at the end. Yeah. Thank you, both of you, for your nice presentations. Thank you all the participants for joining. I hope we could interest you in the topic of sustainability in industrial biotechnology. I want to announce the next webinar, which will be in a month, in Monday, May 26th. The topic will be artificial intelligence. The next time, Lukas Madl will actually explain why we need to integrate ethics into AI-assisted research, and Christian Gruber will discuss the promises and risks of AI-generated synthetic data. Yeah. And if you are interested in talking about modeling and simulation in the framework of this webinar, please write us an email. We are always open for new speakers in this series. Yeah. And as you can see, we have breakout sessions, breakout rooms in a separate Zoom meeting now. If you have further questions for Barbara or Peter, the link for that is in the chat. So yeah, please join us there, and otherwise have a nice day.
See you, and thanks for you.